Hello everyone, I am Dr. ShudhKirti, Assistant Professor from Zoology Department at Sri Sai University, Palampur. Today we will explore an essential technique in biotechnology and molecular biology, that is electrophoresis. This technique is a foundational in separation and analyzing biomolecules like DNA, RNA and proteins. First of all, what is electrophoresis? Electrophoresis is a laboratory technique used to separate charged particles based on their mobility in an electric field. It is a critical method for analyzing the components of complex mixtures, whether you are working on with nucleic acids or proteins. Principle of electrophoresis. The principle behind electrophoresis is simple, that is, charged molecules migrate in response to an electric field. The rate of direction of this movement depends upon their charge, size, and shape of the molecules. Smaller molecules will move faster, while the larger one moves more slowly through the medium, which is usually a gel matrix. Types of electrophoresis. The first type is agarose gel electrophoresis. Agarose gel electrophoresis is widely used technique for separating DNA fragments based on their size. This method, DNA samples is loaded into well of a gel matrix made of agarose. An electric field is applied. The smaller DNA fragments migrate faster through the gel while the larger fragments move slowly. Applications. It is widely used in DNA fragment fingerprinting and analysis of PCR products. Second is polyacrylamide gel electrophoresis, that is PAGE. It is offer high resolution separation of proteins and smaller DNA fragments compared to agarose gel electrophoresis. It is commonly used in biochemical research for analyzing protein samples. In PAGE, the gel concentration can be adjusted to optimize separation based on size and characteristics of molecules being analyzed. Applications: Western blotting and protein analysis. Third, capillary electrophoresis. It is a technique that utilizes narrow capillaries filled with conductive buffer solutions. The charged molecules such as DNA fragments and proteins are separated as they migrate through capillary under the influence of an electric field. The capillary electrophoresis offers rapid separation and high efficiency, making it suitable for various applications. Applications of this electrophoresis, that is DNA sequencing analysis of pharmaceutical compounds. Fourth, pulse field gel electrophoresis, that is PFGE. It is a specialized form of gel electrophoresis used to separate large DNA fragments. It employs alternatively electric fields that periodically changes direction and allowing for the separation of fragments larger than those typically resolved by standard gel electrophoresis method. Applications. It is typically used in typing bacterial stains or and genomic applications. Fifth, two-dimensional electrophoresis, that is 2DE. It combines separations techniques. First is isoelectric focusing and second is SDS page to achieve high resolution separation of a complex protein mixture. In this, in the first dimension, the proteins are separated based on their isoelectric points, followed by separated in the second dimension based on their molecular weights. Applications: proteomics research and analysis of protein expression patterns. Applications of electrophoresis. Electrophoresis can be used for various widely used applications. First is DNA electrophoresis and protein electrophoresis. In DNA electrophoresis, the DNA fingerprinting and PCR product analysis can be done to this electrophoresis technique. For the protein electrophoresis, the Western blot protein and isoelectric focusing can be done from this electrophoresis. In the clinical application, the diagnosis of various genetic disorders or biomarker discoveries can be done by this electrophoresis. Electrophoresis techniques in research. The electrophoresis techniques used as a co-immunoprecipitation and pull down assays are used to investigate interaction between proteins. It is also used as analysis of gene expressions and proteomics and genomic studies. Electrophoresis in field of forensic and medicine. It is used in DNA profiling in forensic investigation. However, it is also used in diagnosis and monitoring of diseases and pharmaceutical applications. In summary, the electrophoresis is an invincible dispensable technique in both research and clinical settings. It is its ability to separate and analyze molecules with precision has been made a cornerstone in the study of the life sciences. Understanding its principles and applications will empower you to tackle complex biological questions and contributes to advancements in sciences and medicines. Thank you for joining me in this brief overview of electrophoresis.